Hello and welcome to the Enterprise Sessions. My name is Professor Michele Barbour and today I'm speaking with Dr Luke Cox, who is CEO of Impulsonics. Luke, first of all, thank you so much for finding the time in your international schedule to join me today.
Thank you for having me. Travel a lot in this role, but I'm sure academics do too.
To some extent, with probably less glamour. We'll come on to that. But Luke, maybe we can start with kind of looking back. So you're CEO of the spin-out company Impulsonics, but maybe you can tell us a little bit about what you did prior to that.
I have been a University of Bristol person almost entirely throughout my adult life until I started Impulsonics. So I came here as an undergraduate in 2013, which is a worryingly long time ago now. I did most of my undergrad here. I did a short stint at the University of Melbourne on exchange. And as part of that, I got a summer placement working on acoustic levitation in the ultrasonics and non-destructive testing lab in the engineering department. And I found that to be really interesting. I'd also done a little bit of work experience in a building services engineering company. And I realized that while it's incredibly important, it wasn't what I wanted to do. I very much felt like I was working in the realm of the possible, and that was less exciting for me. So I ended up doing a PhD in acoustic levitation and manipulation in the ultrasonics group, looking at new techniques for creating new techniques for being able to control cells and small particles using ultrasounds. And from that, we created this technique called impulse control, which is where the name Impulsonics comes from.
Let me get a couple of things out of the way. When I hear University of Bristol levitating things, I go straight to frogs. That's not where you're coming from, right?
No.
But that's not your field.
Yes.
Not interested in levitating whole frogs, maybe individual cells.
Yes, yeah. The levitating frogs work was done with magnets, so we use acoustics. So acoustic waves are an incredibly powerful tool if you're not able to control the material of the thing that you want to move. And they're also biocompatible as well, which makes very powerful, particularly for biotechnology. Obviously, we have a lot of evidence from decades of ultrasound being used in pregnancy, but there is a regime of ultrasound which is very safe for use with cells. You would not use ultrasound to build a levitating train because you can control what your train is made of, but you can't really control what your cells are made of if you want to manipulate them in biotechnology. So that's why it's such a useful tool there.
Okay, that's really helpful. There's loads there for us to unpack as well. So first of all, let me rewind a little bit. You mentioned the use of ultrasound in pregnancy, and I think most people on the street, if they heard the word ultrasounds, if they've heard of it at all, that's probably the most familiar use of it. So if we sort of zoom out a little bit, that is using a harmless technology to look inside of the body in this case. So that's probably what most people would know of ultrasound. What is it that you're doing with ultrasounds that's not about looking inside things, but actually manipulating and holding and moving things?
Sound is essentially a force. When I speak to you, it produces a sound wave which vibrates your eardrum, which your brain then interprets as a sound. Fundamentally, it's a force. And what our technology is about really controlling how we generate that force. And we use a system that I like to describe as sort of surround sound for cells. Okay. So we use a lot of different speakers to be able to control very carefully the sound that we're producing, and that allows us to move the cells around in various different ways. If you've ever had that sort of experience where a sound goes through your head when you're listening to headphones, you can sort of think of it as analogous to that, but imagine if it moved your head.
So it's a long time since I was a physicist, so I'm dredging around in my memory. But sound is a wave, as you've already said, and it travels through air or solid materials or whatever it might be. Is this, in a sense, a form of three-dimensional standing wave? So you create like a low patch in sound where the thing sits? Is it like a sound box, or am I way off the mark?
It's in that region. So the manipulation we do is a little bit more complicated, but standing waves are a very simple way. As you say, you have static regions of high pressure and low pressure. And to grossly oversimplify the mathematics, things tend to move from regions of high pressure to regions of low pressure. What we're able to do is dynamically move those regions and control them, which gives us a much greater degree of control. So in a simple system, you can control where the cells or particles go. In ours, you can not only control where they go and sit, but also move them around dynamically.
Okay, I got you. So it is a box that can move and can sort of
yes,
adjust them over time. And I imagine when you're trying to do something like this, one always starts with one cell, but can you bring more than one cell into this equation? Can you sort of move them around and manipulate them in relation to each other?
Yes. So our specialism is cells by the millions.
Okay.
Essentially. So we actually investigated trying to optimize our technology to manipulate things on an individual cell level. And we came to the conclusion that that wasn't really a strong area for the subset of expertise and technology that we were working with. So there are other technologies, particularly optical tweezers, which can do that very effectively. Our technology could be optimized to do that, but those technologies already exist and are more mature, but those cannot scale up to cells by the millions. To think of it in very physical terms, the gap in the market that we identified was a physical gap between single cell and really large bulk operations. Is there's actually a lot of manipulation there that people would like to achieve but could not really achieve with current technology.
So straight away I'm seeing the confluence of the research of what you can do and the business acumen of, yes, you can do all these things, but where is there actually a market that wants to buy and pay for the thing that you can do?
Yes.
So tell me a little bit more. Let's maybe stick with just the research that gave rise to this just for a little bit longer. This was developed in a research context.
Yes.
Was it developed with a specific application or aim in mind in its initial genesis?
We had ideas about how it could be applied. And I think we had ideas of, oh, this could be a good idea, this could be a good idea. But the original technology was more speculative in terms of trying to understand what could be achieved.
Okay.
So my original thesis title was Optimal Acoustic Radiation Force Devices. Snappy, I know. But the goal was always to be able to see what kind of complex manipulation we could achieve using relatively simple hardware. That was the goal. And along the way, we began to understand, almost through osmosis, when I was still inside in the starting regions of my PhD, what people wanted to achieve with that. We had ideas about manipulating individual cells underneath a microscope, for example. We did a lot of collaborations through the group on things like tissue engineering, so being able to control cells for that application. But really, the focus of the original research was, can we do it and what can we achieve with it?
What can we do? And then move on to, on the basis of what we can do, what might we best apply it to?
Yes.
You're an engineer by training.
Yes.
And what I know of your collaborators engineers, so why the very strong leaning towards the biological organisms? Was this ever something that you might have used in a more pure engineering sense, or was it always the intention to apply it to cells and biological tissues?
When I came into the work, and I should say that some of the underlying research had been going on for about 5 to 10 years before I'd actually come in, we had very much narrowed down on cells as an area that was particularly interesting. Some research had been done into the ability to pattern carbon fibers, for example, but it wasn't the optimal technology for that because of the size of again, it comes back to size versus what people wanted to achieve. Interestingly, actually, the person who led on that original work now runs a company called Actuation Lab, which makes valves. So they moved on to different...
Lots of entrepreneurial behaviour in that environment. I love it.
Yeah.
So you've got a fascinating PhD project, which is both applied in the sense that you were trying to create a device that could do things and could be used for things, but also quite pure and fundamental because you haven't decided what those things are yet. That's quite an interesting niche, actually. How did you choose that PhD? Was there specific technology what interested you? Was it the potential applications? Was it the supervisory team, the environment? What made you choose it?
It was definitely a combination between the technology potential, where I thought the technology was in terms of its journey, and definitely the supervisory team. So in terms of the technology potential, the ability to move things without touching them, without moving parts. In my first year of my undergraduate degree, I don't know if they still do this, but they got us to build a cup dispenser in the engineering department, and that mainly taught me that moving parts are terrible and you should avoid them at all costs. So valuable learning there. I think that was kind of what they intended to teach us. But I always think of Arthur C. Clarke's novel, The City and the Stars, which talks about this incredibly far future society, but they have this line about the perfect machine should have no moving parts. And your laptop is a lot more reliable than your printer, for good reason. And so there was that. I think the technology potential, what was very exciting to me was through my PhD, I worked with Professor Bruce Drinkwater and Professor Anthony Croxford. But during that original project, I was also working incredibly closely with Dr. Azir Marzo, who created this device called the Tiny Love, which is an incredibly low-cost acoustic levitator, sort of handheld. You could build it out of a kit for under $100. Wow. And it used to be over $2,000 or pounds to build a kit, and you'd have to buy all the pieces separately. So it was a massive reduction in cost, and suddenly you started to see thousands of these things being built, and it was just more reliable and better than a previous approach as well. And I think there was a confluence of technology including 3D printing, including almost the ability to buy things off places like Alibaba in huge bulk that you wouldn't have been able to get otherwise, but meant that we were at a point where suddenly acoustic levitation manipulation, which was a technology we've been aware of for, well, all the way back to August Kunt's tube in the 1800s, really, it felt like in the last 10 years it'd been really starting to take off, but there was still so much to be done. So it felt like there was a lot of potential, but a lot to be done. And that was really exciting to me as a technology field compared to some of the other places I looked at were more simulation based, things like computational fluid dynamics, which I found quite interesting, but I think it's a more mature field and more difficult to leave such a big mark. And I think I always like to do things that feel like they're important and make a difference. And that felt like somewhere where I could really make a difference and have an impact.
If I may say so, having been a PhD student many years ago and supervised many more since, it's actually a really, really good reflection. That sounds so terribly patronising, Luke, I'm sorry, but it's a really good reflection of not just is the project interesting, but where is the field and what role do I want to play in a field? And I absolutely applaud your decision. So you've described your excitement about the technology itself and the development of the field and the applications of technology. We know, because we've already had the spoiler, that this went on to form a spin-out company. Was that part of your aspirations from the beginning, the commercialization of this technology, or did that come later in your journey?
I don't know that me specifically launching a spin-out company was initially on my radar as I really want to do that. I think I was definitely aware of that being a thing that could be done, particularly because my PhD was part sponsored by Ultraleap. So obviously Ultraleap being a big company that spun out of the University of Bristol, I think in 2013, so the year I started actually. So just as I was building my cup dispenser, Tom would have been flying off to Vegas somewhere. So that's definitely something I was aware of. But one of the things that I liked about the research group that I was in was it was definitely commercially minded. So the ultrasonics and non-destructive testing group had commercialized things in various ways in the past. So obviously they've got Inductor Sense, who launched a spin-out company, Ultra Haptics, then Ultraleap was a spin-out company, but they'd also built the TinyLev system, which was a low-cost kit that you could build, which incredibly increased access to that. Their system Brain, which was basically the ability to focus in all positions when looking for a crack at once using a single scan, they had given it away for free. And I always remember Bruce saying, I knew that we had won the battle on that when he overheard someone asking a vendor of these non-destructive testing probes whether it could support total focusing method. And they went, everyone's asking that.
Brilliant. How satisfying.
Yes.
So you're in a very academic entrepreneurial environment already
Yes.
with Ultraleap and Inductor Sense and indeed there's other technologies.
But they'd also worked with much larger companies as well. So I think I was aware of various routes. You know, they'd had PhDs sponsored by companies like Rolls-Royce, for example, as well. So I don't think I saw that as the only route. And I felt like my motivation was I want the things that I do here to make a difference.
That's a brilliant segue into a good question for me, which is you were keen to realise the impacts of this technology that had been developed but you were open to commercialisation or impacts. So what was it that led you and your collaborators to the decision to spin out as opposed to one of those other options?
So I think one of the things that I came to the realisation of over my PhD was that a lot of people were talking about this technology in general, the ability, the acoustic manipulation, being very good for biotechnology, but nobody was really doing it. And if you read papers, you could see 10, 20 years people have been saying this in papers, and no one was really doing it.
Having had a spin-out company myself, just N=1, I look at that now and think, is that because it's really, really, really hard? Because if they're all seeing the potential, but they're not doing it, did that make the hairs in the back of your neck prickle a little bit? Did that make you think, is this something terribly difficult? Or in your naivety, did you think this was great?
I think my attitude was, fine, I'll do it myself.
Which is a good attitude.
Yes. And that probably segues into where we were looking at when we started on a commercialization journey. So sort of 6 months before the end of my PhD, I started looking at that and I started thinking about how we could do it. So I had an idea, and I was actually primarily inspired by a lot of work with Zia Marzo and looking at creating a kit that would enable biologists to use this technology in a very flexible way. So the idea would be there would be some kind of a control box that people would buy from us. It wouldn't be able to be as simple as an Arduino because of the TinyLev, because you still need to create it functionalities, and then they could either buy something from a set of end effectors or they could build their own if they so wanted to. I quickly realized that while the TinyLev was great for hobbyists or researchers where often people are used to doing physics or building their own kit, so it really met that need, it was a terrible idea for trying to get biologists to adopt this technology, because you tell them, and now you get out your soldering iron, and they've stopped listening.
Well, the number of biologists that have all the biology skills they would need to do what they want to do and the skills to build and manipulate and operate that kind of device, I imagine it's quite a niche. It's quite a small overlap in that Venn diagram.
Yes, I very much think so. And I think there is something in there that a lot of people fall subject to this, but you consider the skills that you have to be rather basic and the skills that other people have to be vast and unknowable. You go, well, of course, if they can do biology, they could write a little bit of code or plug a little thing together. And sure, they probably could with the right training and support and things like that. But there's that understanding that they're not perhaps inclined, and it would be very scary to them. And actually, you're building off a much higher level of base knowledge than you're thinking about.
Absolutely. I've seen that time and time again. You're right. You undervalue or just don't recognize your own expertise. I think it's a very human thing to do. Your initial idea to make it like a kit, like the Tiny Lev, you decided quite quickly was not necessarily going to have a sufficient market. The number of biologists that could do that, wanted to do that, was quite small.
Yes, I think the number of people who could have benefited from it if they adopted it was reasonably large. The number that would adopt it was probably maybe in the hundreds, if we're being optimistic.
And did you come to this realization through sitting down and thinking about it, chewing it over with a friend or on your own? Or did you have to learn that the hard way by speaking to the biologists and trying to pitch it to them almost and them looking at you blankly?
I think it was a little bit of both. We are quite lucky in that we had a good collaboration with Dr. James Armstrong, who works down at the Dorothy Hodgkins building here at the University of Bristol. He's one of the small number who will build his own device, but also does live cell biology. I was perhaps skewed by my experience with him, but he had a broader perspective going, but I don't think that's going to
Great. He was the overlap of that Venn diagram. Who could say, do you realise how rare these skills and interests are?
Yes. And I think also from our collaborations with biologists, we were understanding how that sort of world works and the value that we were bringing to it. It was interesting, actually, because this was the first time where we actually accessed any commercialization funding. So we went to the technology transfer office. So I basically put together a little, I wouldn't call it a pitch deck now, but perhaps I would have called it a pitch deck then, which was very helpful because obviously Bruce and Anthony had experience with this sort of world. So they were happy to look at it and give me some initial commercial insights. Okay, you've got to think about these things and those things. And we put together a little bit of a proposal for the technology transfer office. And to their credit, they saw what a terrible idea I was currently proposing was. But saw that there might be the seed of something much better.
They're very, very good at that. Seeing sometimes the nugget of gold buried in the middle. I've seen them do that with others as well. So that's great. So they saw your nugget of gold hidden in maybe the slightly less sort of achievable aim. Were you then successful in securing that funding?
Yes. So we got £15,000 from the EPSRC Impact Acceleration account, and that allowed us to build a very early proof of concept that we could combine the electronics and we could build one of the first kinds of these little units that people could use with our best guess at what people would want. Yep. And then we were able to take that to the Microtask, which is a micro manipulation robotics sort of show. I say take it, this was just on the back end of COVID, so I was actually sat with 3 webcams in the university engineering labs at 2 a.m. in the morning because we were working on West Coast American time, which was not the most glamorous experience I've ever had, but it was very useful in terms of being able to talk to a few people, get some insights into one of the early application fields we looked into in terms of organoids, and showed that we could build something and put something together.
The device that you took to the conference, or the sort of online COVID-y conference, this is no longer like your idea to make a kit that somebody else might kind of construct with parts they've bought. This is now a module, a unit that you were at that point thinking, maybe this is my product. So it's the unit that has the controller, but also things like the software and the platform built in.
Yes.
Yeah, okay, you've presented this at the conference. You started to talk to people about potential uses maybe in organoid handling and so on. Yeah. Where did you go next?
From that, we got onto one of the small iCure programs, the part-time one that ran for about 3 months, where they get you to try and have 30 meaningful conversations with people. So it's the smaller one, and we managed to get onto it on the back of that. We came in with a few ideas on that one, and we obviously got some training there on things like business canvases, but really the most useful thing was those 30 conversations.
I've had my own iCure journey, as I often mention in these interviews. One of the things I found really interesting is the people I thought were my customers turned out to not be my customers. So who did you approach for those conversations? Who did you think your customers would be? And were you right? Because I've already declared that I wasn't.
I was wrong at the start of that journey, and I will be less wrong at the end of that first journey, but moving in the right direction.
Excellent.
So we spoke to a lot of different people. I think we were very interested in organoids in particular at that point, and we got a lot of interest in that. Organoids is still an area that we are interested in as a company, but I still don't think that it's mature enough and scalable enough to be our immediate priority. Yeah. So we were both right and wrong. But some of the critical things that came out, actually probably one of the biggest insights that I think now make a lot more sense of the last 10 to 20 years of people saying it was useful in biotechnology but not being very useful, was the fact that everybody told us it needs to work in a standard well plate.
So this was maybe one of the things that held people back all those years, is that practicality, which is actually quite a tricky practicality, isn't it?
Yeah. From the technology perspective, a lot of the work that people have been doing was easy to achieve acoustically, but not easy to translate into a standard well plate. And one of the advantages of our technology was that it did translate into standard well plates. So if you looked at what other people were doing, they were doing very cool science and it could be applied in very specific areas, perhaps. But if you wanted to apply it on a large scale, you could make stage 8 in a 10-stage process way easier. But in doing so, one had to redesign stages 1 through 7 and 9 and 10. Of course, nobody's going to adopt that. You're causing so much more work. The other thing that came out of that, interestingly, was an emphasis on trying to do single-cell manipulation for the isolation of cells for medicine production. Essentially, if you want to produce a monoclonal antibody, which is a type of antibody which is produced by cells, you engineer the cell to pump out loads of this drug.
The cell becomes a factory, effectively.
Yes, essentially. And that is 8 out of 10 best-selling drugs in 2021 were produced by monoclonal antibodies. Right at the start, you need to guarantee all of those cells have been cloned from a single original cell. So it's a hugely valuable part of the process. So we were quite interested in that. One of the things that I would later go on to learn was other people had also identified this gap, and there was a lot of other technologies that could have meet that need.
So it's crowded and you're maybe coming at it probably later than a few of these other people trying to achieve this. Yes,
exactly. Okay. So you could walk around some conferences and find 10 different people all trying to flog you a different method for their single cell isolation, and some of them were already products that you could buy.
So while you almost certainly could do it, then it becomes, but commercially is it our priority? So the organoid market, quite small and underdeveloped. The monoclonal antibody production market, already quite crowded. This is a bit of a sort of Goldilocks, porridge is too hot, porridge is too cold,
Yes.
Which was the right porridge?
Interestingly, both in terms of sort of like the scale and the size of things, we landed on cell passaging as our beachhead market. So this is the process for the non-biologist. When you're growing cells in a dish, every couple of days, depending on the cell type, they're going to fill that dish and you're going to have to put them into a new dish. If you don't do that, they're going to overpopulate, become very unhappy, and eventually die. It's a very common process. Everyone's doing it all the time, but it's actually a huge faff to automate. So almost everybody is doing it manually.
And has been for 100 years or more. I mean, it's one of those things, I don't spend loads of time in biology labs, but I've done a little bit of passaging in my time, and it's incredibly repetitive and hasn't changed
about
in
100 years.
Yeah, exactly.
It's probably gone from using mouth pipettes to formal pipettes, but a lot of the stuff. It's interesting, actually, because we found our people within biology, or at least the first people that were most useful to us in automation engineers within biology. And as soon as you tell them you're making it easy to automate cell passaging, they are very excited because they understand the pain.
Yes, they see the need. You found that pain point. So what did you then need to do? Because manipulating individual cells, manipulating organoids, which at least are quite kind of, contained, aren't they? They've got boundaries. Whereas cell passaging, you've got a big old washy old petri dish full of cells. So what changes did you need to make to your tech to make it suitable for that use?
It was actually relatively simple for us in terms of our core technology. It was actually one of the easier things that we could do was moving things. So the way that we approach it is we use ultrasounds to help unstick the cells, and then we essentially push them to one side, we hold them with the sound, and then we use a pipette to remove the other liquid, and then you can add new liquid and then put it into a new plate.
So you're effectively holding them out the way, and then other types of automation move the plate and the liquid from beneath.
We come in from the bottom. So the device that we're building at the moment is essentially a box that you put your plate on top of. And then the sound comes in from the bottom. We move the cells to one, we unstick the cells, we move them to one side. You remove your liquid, you add your new liquid, you're good to go. Removing the liquid is something that's a relatively well-established technology. The first product we're looking at is a box which sits inside a pipetting robot. Pipetting robots are a reasonably well-established technology, so we're not building that ourselves as a starting point. Our technology allows you to help unstick the cells much faster than current techniques, and then essentially pushes the cells to one side. You can then remove the liquid, put the new liquid in, so now you've got your cells in suspension, they're called, floating around inside the liquid, which you can then just put into new plates and they are ready to go. And the current process involves often putting things into an incubator. It involves then having to agitate them depending on the cell type. You then have to centrifuge them, and then you have to have a large pipetting robot that can handle all of those interactions, which suddenly means that you've got a machine the size of a small room that requires a lot of optimization just to do this simple process. And people like their automation modular where possible, because it means you can reconfigure things easily. A walk-up device is a lot easier to use and a lot easier to drive adoption within a lab, essentially. So the advantage of our technology is it's very scalable. You can do a lot of things in parallel, but also it can be made very modular, so it can scale from very small labs, which would never currently adopt the automation technology, up to huge labs which want to do massive throughputs and also allow you to do lots of different cell types in parallel as well.
So hugely powerful, and also addressing a real gap in the market where other people have tried and failed, rather than markets where people are trying and succeeding or the market was too small. So fantastic. You've got your beachhead market. At this point, are you still within the university? Have you formed the company? When did the company kind of come to be in this journey?
We had proved some of the technology points using the larger impact acceleration account grant, and then we had gone on to the larger ICUR program. So that was where we identified cell passaging as a key beachhead market, and I knew I was onto something when I kept being introduced to people's boss.
That's always good. Yeah.
So we sort of went with one of the big automation companies. We went from someone on the floor of Medica right up to the person who reports to the CEO, about 4 layers in the space of about a month or something, of people introducing, going, oh, you need to meet and talk to this person.
That's very encouraging.
Yes.
Fantastic. You formed your company. You took the role of CEO.
Yes.
That's a bold and, may I say, very step I very much applause, because a lot of people like you exiting your PhD, moving into a company, would take maybe the chief science officer, chief engineering officer, chief operating officer, chief tech officer, one of the many, and may or may not move on to CEO later, but you went straight into CEO. Talk me through your decision making.
I think I was the only candidate. But I'm not sure I want to say that.
Well, no, I totally get that, but I suppose what I'm getting at is lots of people would just find that too scary. I have sat in this room with people, dare I say, double your age, who went, oh no, I couldn't do that. Oh my goodness, I'm putting words in them. That's certainly how I felt. And I had a great deal more experience than you when I went through my equivalent journey. You laugh and say you were the only candidate, but actually there are people out there that make a career of being a jobbing CEO of a spin out company, which in some ways can be more comfortable for the entrepreneur. They feel like, well, they've got the business. You could have, I'm sure, found someone who was willing to be that CEO, but you took it on yourself. First of all, good for you. But what, I suppose, were you reluctant to give that degree of control away to somebody else? Was it your tech and you felt it had to be you that brought it forward?
I think the honest answer is I was probably the best candidate around at the time.
Yeah.
So we did consider looking for external CEOs. I think we felt for the early stage of the company and what it needed, we needed someone who could see the technology and what it could do and marry that with seeing the customers and what they wanted and how they were operating. And I think one of the things that I found during the ICUR journey was that I was quite good at that talking to people bit. Yeah. I hope that's coming across here.
I was going to say, I can believe that, because I'm seeing it right here. But that link up between the tech, which is often where the new founder feels more comfortable, because that's often where they've come from, is the tech or the science or whatever it is for them. That talking to people and talking to people in a non-technical language, or indeed in their own technical language, which might be just technical language different from the technical language that you grew up with, that's something that doesn't always come naturally. But it sounds like for you, it's something you sort of slotted into quite naturally, which is amazing.
Yeah, I think there was definitely something in that I had been learning the language and science of biotechnology for 2 years, jumping in a long way at the deep end, from going from my GCSE at biology all the way up to conferences where PhD and post-PhD people are presenting their work. So I spoke and understood biology on a certain level. I wouldn't claim to have a deep understanding, but I had an understanding on the right levels, and I understood our technology and what it could do. So I was in a very advantageous position there, and I don't think that there were any particular candidates who could see both aspects of that clearly. So I would say there are definitely, as you say, a lot of jobbing CEOs. I think we were a slightly unusual company. For example, there are a lot of companies in biotechnology that are working on new therapeutics. And that is, while not an easy path, a reasonably well-defined path. There's a reasonable expectation of you're going to need to take it towards clinical trial, de-risking the science at each stage. Exactly, yeah. And I think you could probably find a very competent CEO for that. If I had been running that kind of a company, I think I would have been a bad choice for CEO.
Yeah. Because there's a template, and ideally you want someone that's been through that process before with previous companies, so they kind of can use that experience and those contacts and networks. But in this instance, it's more of a one-off, or at least something more novel, a less clearly defined path.
Exactly. Yeah. So I had a good sight on the technical aspects, I had a good sight on the market aspects, and I had a good sight on the application, and I could speak all of the languages necessary to a sufficient degree that I could act as a translator between them and treat things as black boxes where necessary and trust people to tell me when that was now a problem. I think it was also in part a fact that we got a very good engineer who I felt I could trust to really focus on the technology.
Yeah. That freed you up to not be doing. You could understand the technology, but you weren't actually doing it.
Yes, exactly. And I was still very interested in it and still contributing to it. And still, the other day we were talking about mechanisms for a new technique we were trying to develop. And that was still something that I'm able to contribute scientifically to, I hope. No one's told me I'm wrong yet. No, people tell me I'm wrong all the time. No one's told me I'm useless yet.
Okay, let's hope and believe that day will not come. Yes. So what year are we now? The company's just formed. How long ago was it?
So early 2023 was when we went through the iCure options roundabout. So that's where you present your idea. We said we want to work on the cell passaging automation, and then we were able to apply for the iCure grant. We were very lucky. We were the only one of our cohort to get out on the first try. So November 2023 was when we kicked off the company in earnest.
Fantastic. And so one thing that all company founders discover, some of them expected it, some of them don't, is once you have a company, things cost a lot of money. And I actually think genuinely, like as researchers, we know what our reagents cost or what that microscope costs, but we don't always fully digest, despite a university's best attempts, to really see that the complexity and the cost of a proper organisation, even a really small one. So what I'm saying is companies need money. So you got some funds from out of the iCure program at the end of the options roundabout, and you got a grant. Was there other things you needed to do? Did you need to look for other grant money? Did you look for investment? Did you look for, charitable grants? Or did you manage to bootstrap it on just that initial grant?
So we had that initial grant, but one of the really important things that I think they only tell you right at the end, and they really ought to tell researchers earlier because it matters when you're spinning out, is that no grant is ever paid in advance.
Oh my goodness, what a nightmare that is. Yes. How are you supposed to pay your staff?
Yeah. Exactly. So everything is paid in arrears, which I think is a real systematic problem on the part of the UK government that enables rich people to take more advantage of grants. So we managed to get some money from the University of Bristol Evergreen Fund. Yeah. We also managed to corral some angel investors. So that year, I'd also won the New Enterprise competition, and one of the judges on that then went on to become an angel investor alongside some of their other connections. And then a little bit later, we managed to close that pre-seed round for about 450k.
That insulates you against these grants paid in arrears, that you've got you've got liquidity.
Exactly, yes. But it's very much a stressful experience, I think, thinking we've got this grant, Innovate UK is leaning on you to spend the grant as soon as possible because they don't want it to run over their financial years. And of course, they will not give you the money in advance in case the funds get misallocated, which is laughable given, I think, what we know about what happened in COVID.
Absolutely. And I guess, in a sense, the Innovate UK grant, I'm not going to say it de-risks it for the investors, but it gives the investors confidence, because those Innovate UK grants are not easy to come by. There's a lot of research and diligence they'll have done on assessing that application. So I would imagine for the angels, they're looking at this going, well, I know there's something here if Innovate are putting in this big chunk of cash.
Yeah, exactly. Yeah, so it definitely de-risked that. I should also say the founders as well, we put in some of our own money and it wouldn't have been possible without that. And again, that's another... It's another barrier to participation in that kind of innovation journey, I guess.
It is, and it really narrows down the paths available to people who aren't in that fortunate position. I absolutely agree. So you formed the company, you've obtained funds to support it. What next? What were your priorities? What did you need to do?
So the main priority we had was turning it from something that worked on our benchtop and we'd sort of done some of the basic proof of principles into something that actually looked like the real product, so to speak, and could perform the whole process end to end. So we'd sort of proved some things on single wells and on smaller pieces of hardware, but we did not work reliably. My GCSE electronics designed boards, literally my design, I have a GCSE in electronics, that was the level it was at, was not good enough, so we needed to upgrade that. We needed to sort of do some deeper validations as well and actually optimize those processes. We knew where we wanted to get to and we had confidence that we could get there, but there was still a lot of work to get there, and obviously developing hardware takes a lot of time. Doing life cell biology takes a lot of time.
So it's primarily technological sort of challenges and milestones you talked about there, including doing your own biology. So you weren't sort of having, I don't know, somebody else supply the cells for you to work. You actually brought that in-house.
No, we have a life cell biology lab, which we run up at Future Space.
I know it well. Yeah.
I've realised now we're actually relatively unusual, even among companies aiming to automate cell culture. I have spoken to companies with multiple millions of pounds worth of seed funding, who their entire thing is automating cell culture, who do not have any live cells in their lab, which is extraordinary to me.
And quite possibly do it with polystyrene beads or something. Yes. I'm less surprised than that, but only because I learned it the hard way as well. You've already made really clear that this kind of cell manipulation for passaging is ubiquitous. Like any cell biology lab will need to do this. But you've also pointed out there are tiny labs with just a couple of people, and there are vast industrial labs and everything in between. At this stage, are you targeting a particular segment of that market? Are you going for a particular size of lab, or have you identified where the need is most urgent, or indeed where they can pay for it?
Yes. So our current target is actually on the larger automation systems. So we found the best educated customers, shall we say, are those already working within automation, because they understand that this is an extremely common process. They understand that it's difficult to automate, and if you could make it easier to automate, that would turn the top branch fruit into low-hanging fruit. So they have a strong understanding of it, and often within large pharmaceutical companies, for example, they have groups of automation engineers whose broad mandate is automate things within this company.
Yeah, okay, I see. So it is the big, large companies with their automation divisions within.
Yes, yeah, very much so. But as part of that, in order to focus our technology development, we're also working quite closely with the liquid handling companies. So our first product, we intend to sit within existing pipetting robots, and they're quite excited about that possibility of opening up a new, not a market segment they couldn't access before, but one that's very difficult for them to access before. So those are the people we've really found understand the problem and like the first solution we can get. So that's sort of our focus at the moment, and that is something that we also iterated over the progress we've made since we sort of launched the company, because you never really stop talking to your customers during that process. So we've sort of gone from, we think we can do this thing, here's a little bit of data that we've got, to, okay, we've generated this much more robust data, what do you now think, how would you like to see it? And you often find that you need those proof points in order to deepen the conversation and get more information out of people and more engagement with people. But as long as they see motion, they're happy to give you some engagement. And yeah, we've been figuring out what our next stages of testing are as well. Obviously, there are millions of different cell types. We can only work on a certain number at a given time. So being able to prioritize those, as well as something we've communicated with customers with as well, and also understanding what the exact specifications of a final product need to be, and asking them, no, really.
I do think that's a skill that has to be learned and developed over time. And it's certainly something I think a lot of early entrepreneurs don't appreciate. Is it's easy to get a polite answer to when you pitch your technology that actually masks, actually, no, this isn't what we need. This isn't what my company would invest in, or it's all very nice, but we wouldn't pay for it. It sounds like you've developed a relationship How have you achieved that? Because that's not easy to do.
Yeah, so I think on one level it is once you have found something that people actually do and want. I knew I was onto something when people kept introducing me to their boss.
Well, yes, yeah.
But I think there is definitely a sense of when people want something, they'll put work into it. And I've heard some salespeople talk about it as making them feel some pain. And I guess on some level that's true, but I think it's when people want something, they will put work into making it happen. And that is really the measure of it. If you're able to follow up with people and they want to respond, and sometimes you have to, people will be honest, like, yes, I would like this, but I can't engage at this point. Please come back to me when you're at this point. And that's a helpful traction point. And we have found there people who said, yes, get back to me when you've got this thing. When you come back, they do continue to engage in you. So there was definitely something there. I think there was an important question that often people don't ask, but is an important one, which is what would stop you from adopting this. And actually, if you try and push people a little bit, you can gauge from people's answers there what their actual interest is. Because sometimes if you say, what would stop you from doing it? And people be like, oh, if it was too expensive, I wouldn't do it. That tells you that there is a price point that they are willing to pay. And if the user interface was really bad, for example, is one that we hear a lot. That shows you that they have value. It's very interesting, actually, when we've talked to academics. This is another important point, is academics are often terrible early adopters of new technologies because their incentives are to do the thing that nobody else is doing, which is not a very scalable business model. But we've sort of focused on something that everybody is doing, and when we've talked to academics, they would go, oh, well, no, I couldn't possibly automate that because I think it's a core skill that I think my PhD students need to learn. Of course, when I was a PhD student, I had to mix all my own reagents, and now I just buy them off the shelf. Of course, when I was a PhD student, I had to do this, and now I just do that. And so you realise, oh, the answer is you will adopt this, you're just going to be last in line. Find the person who's got a KPI or something. Like, really interestingly, some but not all pharmaceutical companies, for example, have KPIs related to plastic waste. So if you say we can reduce your plastic waste by 50% in this process, suddenly they're immediately thinking, in their mind's eye, for a second, they are in their performance review and they're asking what you've done against your KPIs, and they're writing it down. They're going, we bought this machine and it's 50% plastic reduced.
That's really interesting. And I would imagine that was not what you thought was going to be one of your core technology offers when you started out.
No.
Yeah, that's fascinating, and valuable insights and transferable to other sort of applications as well.
Yes.
Barely 18 months old, your company, Impulsonics. Where are you now, and what does the next 12, 18 months look like?
So we're currently at the stage where we've got a machine in a box, which essentially works, but is definitely something that sits within our lab and is not something that we'd be able to put into other people's labs. So really the next stage, but what we're looking at the moment, is being able to bring up that technology to a level where it can work into other people's labs and actually start getting it tested within integrated systems. So we've already got some people who we know are very interested in doing that, and we've sort of aligned those, this is what it needs to look like, and now we just need to do it, I guess.
I guess so, yeah. Exciting, though. And you've got partners lined up as well, which means that's not your barrier. In the broader sense, you talked about your excitement for the technology and making an impact in your field when you selected your PhD. If we went forward 5 or 10 years, what would you be really satisfied if the company and the technology had achieved? Where would you like to see it make?
I would like to see a box with our technology in it in every hospital in the world. Yeah. And for me, I think one of the biggest areas that I'm really excited about our technology is the ability to culture multiple cell types in parallel, which enables what's called functional personalized precision medicine. So essentially testing different types of drugs on particular patient-derived cells in order to see which one is going to work best for them.
For that individual person.
For that individual person.
Some kind of anonymized cells, yeah.
Exactly, yeah. So the way that drug discovery currently works is they will test it on different cell types and they will see which is likely to work and they will bring it into clinical trials. And there's been a lot of work done on trying to use genomic sequencing to be able to identify what the best candidates are going to be. And the review I read recently said that depending on the exact disease type, we're looking at about 10% of people being able to benefit from that at the moment. the early studies into precision functional medicine already showing 80% plus success rates. Okay. And you've got to think as well, we've probably had 10 to 15 years worth of billions, let's be frank, investment into this genomic sequencing. And I think there is something that nearly every cell in your body has the same DNA, and yet we do not exist in bodies of single cells. So that basically is screening things for a personal cancer, for example, but also screening to make sure that the toxicity profile is not going to be worse for you than other people. So doing all kinds of different things as well. And being able to grow lots of different patient cells in parallel means that that becomes a much more scalable approach.
I suppose in theory, one could do this now with manual passaging techniques and similar things, but it's completely unattainable because of the scale that would need to be done. So this automation could make it much more in the horizons of what could be achieved.
Exactly, yes. And there have already been trials doing it with manual processes, but I think you'd be sort of starting up huge schools of lab technicians if you wanted to roll that out across every hospital, and to be frank, the economics probably would not stack up.
I can well understand that. Luke, you've had an extraordinary journey in such a short number of years. You've been an engineering student, you've been a PhD student in an interdisciplinary project, but where the cells are sort of provided to you. Then you've formed a company, and then have I missed a bit?
No, I was going to say, if it's for PhD, we didn't get the cells provided to us so much.
Let me do that bit again. I'm going into your personal journey. You've had such a journey over really quite a short number of years. You've gone from an undergraduate here in the university studying engineering. You've done your PhD working on this device that has much promise, but at that point, not yet a really specifically designed application. You've explored a whole bunch of applications. You've taken cell biology in your stride. You've become a CEO, and you're out pitching to investors and pitching to Innovate UK and all this in quite breakneck speed. How has that felt to you? Because that's a lot of skills in a short number of time. How is that?
When I reflect on my life, I think that I do not enjoy being good at things. Which is to say, when I get good at things, I think I'm always looking for the next challenge. When I worked for a building services engineering firm, I was sat down by one of the engineers who sat next to me and had sat in that office for 30 years, and he was like, you can come back here after your degree, and then you can get this qualification, and then you can get this qualification, and you get this qualification, and at the end of 30 years, you can be sat right the desk opposite you.
And to some people, that's a really attractive, stable, known, specific career.
I saw a carpet rolling into my own grave. I think there is something about the next challenge, about doing things which are difficult and exciting and ambitious, that really attracts me. And I think doing my degree, I found that really tough at the time, but I found it very rewarding. And then I found the idea of doing research, knowing things that nobody else knows, only for a brief moment until you actually tell them. But, there's something very exciting about being the first person in the world to do something or know something. That's hugely exciting. And then going, well, actually, I want to do something. I've always been an engineer. I like building things, and I don't think I'm interested in knowing things for the sake of knowing them. I want the things I do to matter. Yeah. And so going on that journey, I guess, has been exciting, exhilarating, stressful, hugely stressful at times. Let's not pretend it's not.
Well, but it's really interesting because we've been talking for however long now, and that's the first time that's come up. A lot of people, particularly entrepreneurs, who often talk about how scary it is and how intimidating it is and how much they felt like overwhelmed by it. And actually, what I hear from you is just this excitement and this joy and this anticipation of the next big challenge. Do you recognise that, or am I describing something that you don't agree with?
I think that does make sense to me. I think I really do love learning new skills, but doing it in a very practical way. And I think this is definitely an opportunity to continue doing that. But as I say, it's always got to be making a difference to some extent. And I think there's something as well, but definitely there are times when it is extremely stressful. I don't want to pretend otherwise from that. I think there's also an aspect about it as well that our chair, Steve Kitson, has a really interesting insight about an entrepreneur not necessarily being a risk taker, but a risk engineer.
Oh, I like that.
So you don't have to take the biggest risk possible at any given time. If you can take a smaller risk, which will then still move you in the right direction. One of the things that's really insightful that he pointed out is, yes, there is a financial and time commitment risk from doing a spin-out or startup company, but there's also another aspect to it, which is the skills that you build. Yeah. And it's very difficult to do a journey like that and not learn a damn thing. And so I guess you have to have some, call it confidence, call it arrogance. There is a risk to doing this, but if it doesn't work, you will find something else equally exciting and interesting to do. If it's worth doing, it's worth taking a risk on.
But I love the concept of, I'm going to add precision risk engineering. That's a really nice way of looking at it. You don't have to take the biggest possible risk. That's fantastic.
There's also luck engineering as well. I'd like to point back, well, I think you don't get to decide where the dice land, but to some extent you can often control how much you roll the dice, and often the cost of rolling the dice is quite cheap. I actually think the ICURE program is sort of built around this idea. It says go out and have 100 conversations, and that 100 conversations, a lot of them will not teach you anything. Some of them will just teach you you're barking up the wrong tree, which is valuable, but some of them will become extremely valuable. And actually, by having some kind of a metric, it forces you to go out and roll the dice a lot more. From my experience, I turned out to be in the wrong room in Boston, having flown out to Boston just for this 2-day conference to learn I was in the wrong room. But I talked to one of the most valuable people that we ever spoke to, who now continues to give us some advice. But 3 months later, I was in San Diego in the right room, and I wouldn't have been there if he hadn't told me to be in that room. And I wouldn't have spoken to the right people if he said, oh, I didn't realize you were here. Let me introduce you to this person from a big pharmaceutical company.
Which makes the room in the Boston the right room after all.
Exactly,
as it turns out. But now you've revealed that you like to do something new and learn new skills. In your words, you don't like to be good at things, although I would challenge that. When Impulsonics is a huge success and is acquired or exits or whatever, which I have no doubt it will, what next? Would you do this again? Would you do another spin out? Or is it like, well, I've done that then, so I'll need a new big challenge?
Not knowing what's next is quite interesting to me. I think on the days where it goes really well, I can't think of any better job. On the days when it's really hard, I would love to walk away and do something completely different, write my political theses or something like that.
Oh, we've got a little glimpse there. Well, whatever it is, I have no doubt it will be something exciting and bold and probably unexpected. So you'll have to come back in a few years' time and tell me about it then. You've had that whole journey from undergraduate, postgraduate, through to wonderful work you're doing in the company. If you were to be speaking to researchers now, maybe people starting off their PhD or in the early phases of their PhD, so they haven't probably crystallized their full plans yet, or hopefully keeping an open mind, what sort of advice would you give them?
The main thing for me is working interdisciplinary and also recognizing that the skills that you have there might actually in academia be way in advance of what is actually the biggest wins in another industry. And that interconnection of academics love to do the most advanced, most complicated, most complex, most difficult thing, which is brilliant, and that is fantastic, and we really need that. But when you step across, you often find that actually, oh, it's not the most complex thing that is going to solve the biggest problem that we have right now. It's this much simpler thing that you can achieve, which is often something that you might be dismissing or thinking is simple.
Yeah, think is a little bit humdrum and well, a bit like you thinking biologists can use a soldering iron. But like that sort of not realising where other people are coming from, where they're starting from in their skills, or what problem they've got that needs to be solved that you can help with.
Yeah, exactly. And also just thinking laterally about it, because I've realised the core of our technology is being able to move cells to one place and move liquid to another place. There's a huge amount of what we do at the moment, which seems obvious in hindsight, but it's not obvious that there's not a great solution to that. A lot of the pain that we solve for people is just that it's annoying to have a large rotating centrifuge mass and the same system as your highly calibrated optical system.
Yes, because centrifuge is like, for those that don't know, your washing machine is a centrifuge, and we all know how the kitchen floor vibrates when that's going off.
Yes, exactly. So it's odd. But also there's a recognition that talking to different people, you might explain things in different ways, and that will make a huge difference to how they understand it. I think there's also something of you don't have to give up the dream or the vision of the big thing, because I would still really like to take Impulsonics to those really big, ambitious, complex things.
Yes.
I just realised that 90% of the hardware we need to build will also solve this problem that people have today, now, and they're lining up. So let's solve their problem first.
It's a little bit about having your eye on the point of the horizon that you want to get to, but recognising there's lots of different routes between here and there.
Yes.
And actually the one you thought of first may not be the one that's most likely to lead you to success. Luke, clearly you've gained a huge amount from your commercialization journey and you've made great progress, but were there any barriers or difficulties you faced along the way? And I suppose from my point of view, what can I and we back in university learn about that to try and remove those barriers for future entrepreneurs?
I think one of the biggest barriers and difficulties that I faced, particularly when I was coming out of my PhD and before we launched the company, was actually the insecurity. So there was a period of about 2 years where the longest amount of job security I had was 6 months, I think.
That's hard. That precarity is excruciating. Very, very difficult.
Definitely, I felt supported by the people that I knew within the university to try and access the resources that were available.
Sure, but they're finite.
The resources were finite, and also the nature of the resource allocation was a progressive de-risking technique on the part of the government, but in de-risking that, they then pass the risk on to the individual.
Yes, exactly.
And I think the other thing is that people do not do their best work when they're worried whether they're going to have a job in a few months.
Absolutely.
And, there were times where I was also only working on this 50% of the time I was paid by the university, 50% of the time I was working for another company. Really, I was working sort of 70% of the time on this and 50% of my time with another company. So there's definitely something there, and I think it creates a structural barrier where I was lucky that I could deal with that risk. Other people would not be able to do so, and I think there's a real problem there that, from my experience, I have been totally disillusioned by the notion that our current system enables the best technology to make it out into the world. I don't think that's true at all. I think we get fantastic technologies out. Probably the system is effective at filtering out bad ideas, but I think it's also that filter is hitting a huge amount of good ideas, not on the quality of the idea, but on the position of a person who happens to have the expertise to be able to implement that idea relative to the position of the funding. And I think that's a huge shame and is hugely to our detriment. So it was odd when I started a company with 18 months of runway when we launched it to suddenly have the biggest job security I had in years.
Yeah, it's a huge barrier to inclusivity in the whole world of entrepreneur, isn't it? In your time over the last number of years, you've worked obviously in your own company labs, Impulsonics, but you've also worked in research labs in university. How different or similar are those two environments? You've done them really quite close together, so compare and contrast.
So I think working within a company lab, one of the things that I found is that we do a lot more risk management and iteration on the technology. So I think it is relatively rare for an academic to really sit down and seriously go, here's the big risks of this project, and here's how we're going to progressively mitigate them so that we're tackling the biggest ones earliest on, so that we've got the most resource for them, and also being able to iterate and go, actually, we don't need to try and build the whole thing now. What we're aiming for is this thing, which might not be the full thing, but we think that we know enough there that we can iterate and iterate and iterate and get there. Let's solve this problem and this problem and this problem. Interestingly, actually, in academia, you often found yourself going, what's the most ambitious version, and then working your way back. Whereas in industry, you're more working towards the minimum viable product is a software term often used. I don't think it's necessarily as applicable as hardware or in biotechnology, but there's something about the minimum viable proof of concept, for example. When you're looking in academia, for example, you might generate very high level of data to be able to publish. In industry, you might generate lower levels of data that give you confidence that you can move on to the next level. If you want 100% of something, you might try and get to 80% first and then see if that works, and then move on to another big risk rather than trying to get onto 100% on that one if you think there's a reasonable expectation that you can come back and get 100%. And it's also about framing your goals relative to what the person wants to achieve. One of the big things that we've learned is that in some labs, they are throwing out 50% of the cells, because they just want the cells to be alive when they're ready to go. It's not true for every lab, but that realised, okay, actually, for some people, they want the quickest, quote-unquote, dirtiest way of doing it. They don't care what their recovery is, so we can make something for them that works even faster.
And if some of the cells don't get transferred, it doesn't matter.
Exactly,
That's not what they
for
care
someone
about.
else, we need to optimise in a different way. They're going to want 100% retention of their really valuable cells, in which case that's a different optimisation we can still do, but there's a trade-off there, and you're iterating both the business case and the technology in tandem. So understanding what the real core of someone's problem is, and it's often not what they think it is, but you have to listen quite carefully
to almost tease out what it
exactly
what's really behind it.
yeah.
Yes, yeah.
I think that's a way that you work very differently, and academics often set their own goalposts, whereas in this you're trying to iterate your goalposts and your technology in tandem with your customer feedback.
What part, if any, does AI have in your company's strategy? Is this something that's useful to you, or is it something that's just not really applicable?
For us, we can help in feeding data into AI. Every large pharmaceutical company is interested in using AI to mine data from their existing experimental results. One of the realizations they've come to is that actually a lot of the data they've stored is not of good enough quality to feed into your model. So you're essentially feeding in noise. I talked to someone from a big pharmaceutical company who was one of the senior technology people just over drinks at a conference in 2023, and they said, oh, the data from 10 years ago, we can't put that in, that's just noise. I talked to someone earlier this year, and they said, well, the data from 5 years ago, it's not good enough quality, it's just noise. So at the current rate, things are looking good. I think one of the difficulties we faced is that AI is sucking up a lot of attention in the investment ecosystem.
So I wonder that might be an impact,
I think there's definitely some real applications of AI. I'm skeptical of some of the others. I was at an investment review where they talked about SaaS was a big thing, fintech was a big thing, and AI is now a big thing. And you go, well, hang on. Wasn't there the blockchain and the metaverse? Why are we not talking about blockchain and the metaverse? Now, I think AI has more utility than blockchain or the metaverse. Yeah, no, I think having like the feedstock is good. I think it's just hugely important to have real data feeding into the AI. We have to be careful about how we use it because it's a probabilistic model. And so sometimes it can be very helpful if you're doing a first pass on a grant application. I tried out one of the deep learning things to say, what kind of a application could we make for this? How might we fit into the scope? And it gave an interesting first pass of it, and you went, wow, that would be a fantastic thing. But I don't think it's possible. It was completely impossible. And actually, when you ask it more detailed questions, it does confess that it's impossible. Yeah. But I think you have to know something about the field. I also get a million AI emails, so I'm very skeptical of AI marketing.
Yes, I can identify with that. I have to ask you one more question. You are the graduate, the PhD, the CEO, the entrepreneur. How many different sort of strings to your bow are there? I found out recently you also do improv theatre.
Well, I found out recently you mean the producer ratted me out.
Yes, okay. I found out in the last 10 minutes that you do improv theatre, which I have to say both surprised me, but also having spoken to you for a little while makes quite a lot of sense. So tell me a little bit about that and what, if anything, what utility that has or how you've used those skills in your commercialization journey.
So I think communication and storytelling are key to any kind of commercialization journey. I think definitely sort of like being able to communicate with people effectively is hugely powerful. Obviously, if you want to do a spin out, you're probably going to find yourself pitching, not just to investors, which is a classical one, but also on some level pitching to potential customers. And that's also where storytelling comes in, because particularly in deep tech, which is often what comes out of universities, you will require a significant amount of capital or investment or grants to be able to realize your vision, which therefore in the current system means that you have to therefore have a huge impact and a potential for financial return, particularly in terms of investment. So being able to tell that story of how the thing that you're developing now weaves into a future, hugely impactful, important, both valuable for society, but also capable of generating a financial return. And I think generally, something that has a large impact in society is capable of sustaining a business model that can support venture-backed companies, like most deep tech companies are. But being able to link those two together and compellingly tell that story and really iterate on that story is really useful. I also think for improv in particular, you spend a lot of time training yourself not to be afraid of looking like an idiot. And I've made a bit of a career being unafraid to look like an idiot. Both in terms of improv comedy, but also being able to, when you're trying to understand something like biotech, ask stupid questions. And just going, this is probably a stupid question, but is often a very powerful way of getting answers, and people appreciate that. But also, there is sometimes something that you have to put a proposition to someone, say, I think this, and you might not believe that, but you need them to tell you if you're right or wrong.
Yes.
People like to correct you. I'm sure there's an internet law about that as well. being able to effectively communicate, I think public speaking, definitely being unafraid of that, having some confidence in that, but also being unafraid to look like an idiot. And
I love it. A lack of fear. We could all do with less fear.
I didn't
Let's
say
more
lack
improv.
of fear.
Okay. A reduction in fear.
A tolerance for fear.
Okay, yeah. An embracing of fear.
Yeah. Scared but did the thing anyway. And often you realise, I think in improv they have this concept of a happy fail, which is that the stakes of failing are quite low. And particularly in something like iCure, the stakes of talking to someone and asking them, do you think this idea is good or bad or not? Well, actually, it's that risk luck engineering thing again. The objective stakes are they might tell you that your idea is bad and then you'll walk away. The potential upside is enormous if they go, that is exactly what my company's been looking for, how can we possibly work with you, or let me introduce you to my boss. Yeah. But the perceived stakes are, what if they don't like it, what if they don't like me, and you're going to feel quite bad about that because you will feel like you've made a mistake. So being able to look a bit past that and go, well, I might make a mistake and I might feel a bit bad, and that's okay. Yeah.
Thank you, Luke. I have enjoyed our conversation enormously, and I've learned a lot, both about you, about your technology, and about your company more broadly. So all that remains to say is thank you very much indeed for being so generous with your time and your insights.
Thank you for having me.